Hey guys, today I am going to discuss a very important topic, that is drugs usage in animal husbandry and environmental impact. The story begins penicillin was discovered in 1928 by Alexander Fleming, who found that antimicrobial properties of a substance exuded from Penicillium notatum might have therapeutic value if it could be produced in quantity. Okay, might have therapeutic value if it could be produced in some quantity, sufficient quantity. Now the story is altogether different. About 1 lakh to 2 lakh tons of antibiotics is produced worldwide, and US alone produces about 22.7 million kilograms of antibiotics. In US, 70% of dispensed antibiotics are given to healthy livestock to prevent infections and or promote growth. This is a matter of concern. This practice is outlawed in European Union countries. Now, categories of antibiotics. There are various categories depending upon mode of action. Number 1, those which inhibit bacterial cell wall biosynthesis. Number 2, those which block 30S or 50S ribosomes. Indirectly means that they inhibit protein synthesis. The example of first category include cephalosporins. This is very important, cephalosporin. It is used in therapy, human as well as animal. Number 2, this includes fusidic acid and tetracyclines, etc. They are also therapeutic and subtherapeutic uses. Number 3, those which block replication of DNA. Quinolines, rifamycins, they are used to treat urinary tract infections, some respiratory as well as systemic infections also. And there is another category which disrupt integrity of cell membranes, like azoles. They are used to treat fungal infections. Then there are some which antagonize metabolic processes. Sulfonamides are one example. These are used in humans, aquaculture and livestock as well. The antibiotics in the environment, where do they come from and how they move from one place to another place? And in the center, at the top of a hierarchy, there are antibiotic manufacturers, from where antibiotics move to pharmacies, then human use, then ingestion and excretions, and flushing of unused, topical applications, municipal waste or runoff, then septic tank also, treated discharge also, these finally go to the surface water contamination, and finally they leach down to potential drinking water sources. The second route is veterinary and livestock. This first route was human beings, now the second route is veterinaries and livestock. Livestock feed manufacturers use antibiotics. They, in fact, they treat the discharge, but ultimately that leaches down to the drinking water sources. Similar is the case of veterinaries. Antibiotics are used in livestock. There is injection, and finally excretion. There may be some treatment of manure, then it is applied in soils, and finally they again leach down into the groundwater. This is ground water or water table. They leach to water table, which is a source of drinking water. Then this is a simplified diagram of the various routes of release of human and veterinary pharmaceuticals. This was a pharmaceutical company manufacturing antibiotics. This is taken by a human being or a livestock. Then finally it goes to the soil and then to the water. You can see many. Now human inputs. The most important is excretion of antibiotics and metabolites, which are secreted directly in amount of about 40% of ingested dose. Then we also dispose of the unused antibiotics by flushing them down into the toilet. Then when we apply topical antibiotics, we wash them. They all go to septic system or municipal waste, and they are the breeding grounds for antibiotic resistant organisms. Veterinary inputs. Antibiotics are used in livestock operations since 1950s. They are used to treat infections or to improve growth and feed efficiency. Four types of antibiotic used in livestock operations. Number one is therapy, control, prevention, and growth promotion. Therapy is treatment of frank clinical disease. Control means the antibiotics are administered to a herd of flock which morbidity or mortality has exceeded baseline norms. Prevention. Antibiotics used in animals considered at risk but where individuals do not show signs of disease. And growth promotion. The antibiotics are administered over a period of time, usually as feed additive to growing animals. Veterinary impacts. Antibiotics impact aquatic environment through manure. Large fraction of medicines present unchanged in animal waste. Direct runoff for collection and use as fertilizer. Research in this topic on impact on environment is in primitive stages, though FDA regulation is there for releasing the drugs. Impact. Number one is microbial resistance. There are workers like Anand and Lapara who have suggested that environmental levels have been found to enhance the ability of microorganisms to develop antibiotic resistance. Then shows evidence of antibiotic resistant microorganisms has been found in surface wastewater, groundwater, and drinking water. That means the microbes are becoming resistant. They have been found in wastewater, groundwater, as well as drinking water. Environmental bacteria as reservoir of resistant gene. Then they are transferred from non-pathogenic bacteria to pathogenic. Single cross multi-resistance. Transfer of antimicrobial from nutrient to plant tissue. I want to just give a single very simple example. If bacteria which are non-pathogenic in environment become resistant to some antibiotics, and that gene is transferred from non-pathogenic to pathogenic ones which are infecting human beings as well as animals, then they pose a great threat. Revisit human health concerns. Like methicillin resistant staphylococcus aureus, it is very difficult to treat. First, it appeared as hospital derived infection. In 1990, community associated outbreaks among high risk populations. Today, colonization rates in general population is about 1.5%. Infection account for majority of skin and soft tissue infection treated in emergency rooms. In 2005, MRSA caused more death than AIDS. Antibiotic resistance as human health concerns. Enteric pathogens such as Salmonella and E. coli causing illness and treatment failure. Other multi-drug resistant organisms like Klebsiella, Pseudomonas aeruginosa. Relatively few pathogens in nature, but antibiotic resistance is transferable. How to detect antibiotics in water? Very sensitive low level detection available through GCMS and MS and LCMS-MS. Example: minimum reporting limits: ciprofloxacin 20 mg per liter, and then we have erythromycin. You can see the examples. These are the reporting limits. There is one more very important, which is drug that is known as oxytocin. It is directly related to milk. In mammary glands, there are alveoli which secrete milk. Cistern store the milk. Then milk ejection is stimulated by pituitary gland's oxytocin release. Contraction of myoepithelial tissue, and then milk comes out through a pore which is about 20 to 40 IU. There is 11.6% increase in milk production by utilization of oxytocin. This modifies the milk quality. The milk contains more fat, less protein. High dosage negative effect on fat. The residual oxytocin in milk is a human hazard. Now we come to a very important topic. This is very important. In fact, I wanted to make a separate lecture on this. Somehow, if time permits, I will definitely make a preview of local population of vultures in Jammu and Kashmir, also separate lecture. India has 9 species of vultures. These are white-backed, long-billed, red-headed. We have different names. You can see Gyps bengalensis, Gyps tenerostris. Then Himalayan griffon. The population of 3 species. This is very, very important. The population of 3 species: white-backed vulture, slender-billed vulture, and long-billed vulture in the wild has declined drastically over the past decade. Because of the evidence of widespread and rapid population decline, all 3 vulture species were listed by IUCN, International Union of Conservation of Nature and Natural Resources, the World Conservation Union in 2000 as critically. And this is our white backed vulture Gyps bengalensis. It is widely distributed in India till late 90s. It was abundant but declined rapidly. Cause is veterinary use of diclofenac for livestock. There are various initiatives for its conservation like BNHS, Vulture Conservation Center at Vinjor, Buxan, nationwide ban on diclofenac, vulture advocacy programs, comes under Schedule 1 of Indian Wildlife Protection Act 1972. There are many, no. But what is important, its population declined rapidly after 1990. This is second species which is under decline, that is Gyps indicus, long-billed. You can see it's long-billed. Long-billed vulture Gyps indicus. It is widely distributed in India. It has long beak. Like white-backed vultures, their population declined rapidly after 1990. Cause diclofenac again. The similar procedures of conservation like that of white-backed vultures. Significance of these vultures for human beings. They are scavengers. They scavenge on animal carcasses and thereby clean the environment and dispose of the dead bodies. This is even a religious practice by Parsis. Cause of decline. Captive vultures are highly susceptible to diclofenac and are killed by kidney failure within a short time of feeding on the carcass of an animal treated with normal veterinary dose. Now, whenever we give dosages of diclofenac to animals and even then animal does not survive, it is eaten up by these vultures and they die of kidney failure. There is an alternative drug now that is called as meloxicam. Thoughts. There are lifestyle changes, hormonal changes. Since we utilize poultry meat, and in poultry meat there are growth regulators, growth promoters. Ultimately, we take Ultimately, we drink contaminated water. How to reduce the release? There are some treatment methods like sorption, volatilization, biodegradation, chemical reaction, and ozone treatment. Thank you very much. Have a great time. Please subscribe our channel WebGear 9. God bless you.